of the Climate Smart Agriculture webinar series. What a wonderful day it is in the Caribbean on Double Day. I mean, just setting up to look at Elaine do that double. Today we're in for a dynamic duo. We're into roots and tubers. And a day after Emancipation Holiday, we're into roots and culture as well. Today, we're going to have two presentations by Drs. Robin and Rankin. And for those of you joining us for the first time, the format is simply this. Because I am on a first name basis with both presenters, they have agreed that I can go a morning at 17 minutes into their 20-minute presentation so that they know they have 3 minutes left. We'll have a presentation by Dr. Robin first, then Dr. Rankin, and that will be followed by 50 minutes of questions and answers. My name is Steve Maxime. Welcome to day 5 of the Climate Smart Agriculture webinar series. My task is a particularly simple one because I'm a particularly simple fellow. All I'm going to do now is introduce Dr. Gregory Robin, and we will be on our way, literally, as I said, in this double day. Thank you, Elaine. So, Dr. Gregory Robin is a root and fruit crop agronomist who worked with the Caribbean Agricultural Research and Development Institute, CARDI, for over 35 years. He was the CARDI representative in Jamaica, in St. Vincent and the Grenadines, Antigua and Barbuda, and also in Dominica. Dr. Robin has held the following positions: Manager of Science, Technology and Innovation, Joint Coordinator of the Agricultural Component of the IDB UE MONA Pilot Program for Climate Resilience, which is part sponsor of what we're doing today, Technical Coordinator for the Eastern Caribbean States, and Technical Coordinator for the CARDI Roots and Tuber Crop Program. Dr. Robin holds a PhD in molecular pathology, an MPhil in agronomy and plant physiology, and a BSc in biochemistry. Dr. Robin has also a wide range of experience in farming systems in the Caribbean, and he's been a representative of the Caribbean on the board of the International Society for Tropical Root Crops. Dr. Robin is the author of at least 20 root and tuber crop publications. These include CDs and CD-ROMs and manuals. So without any further ado, I'll just want to turn you over to Dr. Gregory Robin to do his 20-minute presentation. And Greg and I have agreed at the end of 17 minutes, I will give him a 3-minute warning to wrap up.
I need you to share your screen.
Desi is going to give you permission to share your screen, Greg.
Greg, you have permission to share your screen, so you can go ahead.
All right.
We're seeing you.
Can everybody see the screen?
Not? Yeah, yes, we are seeing it now. Can you
All right. Okay. I guess everybody knows me, so I'm going to. I may take off the video during the course of the presentation so that we don't get any distractions. My presentation this morning is on climate smart production of root and tuber crops. I have a short introduction in terms of the format of the presentation, government policy, the characteristics of root and tuber crops, and then I will focus on some climate smart practices with respect to land availability, planting material, soil and water management, very briefly because that was detailed by Dr. Simpson and Dr. Lindsay, soil fertility, production systems, and weed control. The other areas of the production systems like harvesting, post-harvest, and pest and disease management will be addressed in the coming weeks during the webinar series. As we know, the governments in the region have endorsed the root and tuber crops as a key component of the food and nutrition security strategy. And the roots and tubers are supposed to play a crucial role energy per unit area is more than any other food crop. They're rich in dietary fiber, minerals, vitamin B, vitamin E, vitamin C, and antioxidants. The orange and purple flesh tubers are rich in antioxidants like beta-carotene and anthocyanins. They contribute to income of against hunger and natural disasters. The root and tuber crops are found to be adaptable to diverse soil and environmental conditions. Currently, the majority of smallholders grow root and tuber crops in the Caribbean on less than optimal conditions. However, the resulting low yields are well below world average and insufficient to satisfy food and nutritional security needs. For example, sweet potato. The average regional yield is about 7,500 kilograms per hectare. The world average is 11,400. And Jamaica, which is a huge sweet potato producing country, the average is about 18,100. Kilograms per hectare. An average yield of 4,500 kilograms per hectare in 6 Caribbean project countries we currently are working in shows that we're well below the world average. Yam, again, if you look at the figures, it's below the world average, and therefore it's clear we have machine is another root crop, which is probably more focused to about 3 countries, Dominica, St. Vincent, and Jamaica. Those are the other leaves are used as a vegetable, which is very important. And the taro starch has some very low granular size compared to the other tubers. And that makes it an excellent ingredient for baby foods. And I don't think most of you know, taro has 98% digestibility. So therefore, when we're talking about climate smart agriculture where root crops are concerned, we have issues of marginal lands, poor quality planting material, inadequate soil and water management practices, poor soil fertility, inadequate carbon sequestration, and weed control is becoming more and more costly. As I said, the other areas in the production chain will be addressed in subsequent webinars. The present situation, therefore, we have low productivity, cost of production is high. Security. GAP is not fully adhered to. Organic production is not certified where it is practiced. And on top of all of this, we must adopt climate smart agriculture practices in order to survive. Climate smart practices can therefore be viewed as a package of adoption practices, inclusive of all of the above. So because of the interest of time, let's go straight into some of the climate smart practices. Land availability. The agroecological zones for growing these root crops. To view that the appropriate sites for production of the root crops are competing with other commodities also. And in selecting these land sites, the focus should be on the soil type and the rainfall. These are key components of the production system of root crops. Planting material. Predominantly, the planting material is vegetative. And over time, because of cross selection, the planting material becomes of poor quality. There are several ways we can handle planting material, which is the key part of the process, the beginning of the process. tolerant to heat, salinity, and drought. We can get those in, and where available, let us use the local commercial drought tolerant material planting stress for planting in the dry season. Have this improved commercial variety stored in the seed banks. We have the tissue culture laboratories in St. Vincent, the SRC in Jamaica, St. Lucia, Barbados. Disease material and reducing our yields just from poor planting material. And when we put on top of that, poor management practices, our yields obviously low. How specialized farmers produce planting material. And I will demonstrate that in a picture as we go through the presentation. How specialized weeding and hardening facilities, virus mitigated, so we don't contaminate through vectors the planting material from the beginning. And select planting material to satisfy the market demand. This is some work we did under the very PPCR project, and we identified ganja in Jamaica as having drought and soil tolerant genes. Our field research also shows that ganja has significant drought tolerant traits when you compare it with the other genotypes. We have facilities. This is a tissue culture lab in St. Vincent, built specifically for root and tuber crops to store, clean up, multiply, distribute. That lab is compromised because of the take a period of a few weeks to come back to the field. And this is an example of sweet potato, some work we did in Antigua. From the farmer's request back to the field would take approximately 30-something weeks, all right? But this needs to be done to ensure we have a proper productivity coming out of the crop. Now, I put up this slide here to show mini setting in yam. That particular technology came from IITA, International Institute of Tropical Agriculture, in Nigeria. The mini set was never meant to be planted straight into the farmer's field because it's compromised because of cut surface, dehydration, the cut surfaces get infected, if it gets too dry, they dehydrate, and then you lose a lot of your planting material in the field. That mini set was meant to produce small whole tubers that will be sold to the farmers for production. Small whole tubers eradicates all the issues of dehydration, pest and disease, and having to supply in the field. Okay, let's move to soil and water management. I don't have much time for the presentation, so I have to go through it very quickly. Trickle irrigation is arguably the best method for these crops under prevailing climatic conditions, since it conserves water, prevents nitrogen leaching, and we heard about the use of tensiometers, which can be attached to the system. They can be automated and stuff like that. Yeah. Soil and water management continues minimum till. This is recommended for dashing production, as ridging is not required. And I'm using the root crops throughout the presentation to demonstrate this issue. Effective minimum till conserves soil moisture through non-disturbance of the soil surface. It prevents soil erosion, it increases carbon sequestration, and it increases activities of microorganisms because you haven't disturbed the soil surface, you haven't turned it over to expose all the microorganisms to solar radiation, which affects them and so. So you have a more wholesome soil from minimum till. region is small basins that are rectangular in shape are formed within the floor of the ridges, mainly to enhance the storage of rainwater and for allowing more time for the water to percolate. Tide region optimizes water filtration, it improves soil and water management, it enhances and they call it tide region because you build like a miniature mound or ridge to tie this ridge, and this is the basin, and the rainwater mountainous countries like Dominica, St. Vincent, and bridging along the contour will allow you to develop that technology. Mulching. Mulching has many benefits. Weed control, reduces soil erosion and runoff, reduces soil-borne diseases, yielding cassava and sweet potato. That work was done, and it clearly shows that mulching does do that. And it's also been reported that plastic film enhances dry matter accumulation, plant height, leaf area index, tuber yield of potatoes. And plastic mulch conserves soil moisture, which contributes to better growth and tuber yield. One of the reasons I'm spending some time talking about the benefits is because I think in the farming community, while farmers do apply or adopt a minimum amount of technology, they probably don't soil erosion and weeds can be a problem. Therefore, mulching is highly recommended. Intercropping. Intercropping cash crops with root crops, adaptable to the same ecological zones. And when you intercrop, you can't plant crops that are not compatible or not suitable for the particular zone that the main crop is in. And therefore, the root and tuber crops act as a high energy staple for farming, helps to feed animals, acts as an insurance against risks and disasters, and so and therefore intercropping is an important factor there. All right. Integrated crop livestock systems. Livestock transforms plant residues and byproducts into edible, high quality protein and manure, which is an organic fertilizer and increases crop productivity. That should be a must in our climate smart systems. Worldwide livestock integration with crops is the only large scale example of successful long-term integrated production systems at the supply chain level. So it's very important that in our climate smart system, we recycle stuff through the animals and generate both protein and compost systems. Integrated weed management. A simple definition of integrated weed management is to use more than one weed management method, whether it's biological, chemical, cultural, or physical. The basic goal of integrated weed management is to restrict weed growth until a crop is sufficiently well established. So in the development of the crop canopy, all right, during that period, to allow the crop to outcompete the weeds. Now, most of our farmers use herbicides, but I just accidentally visited a farm over the weekend where the farmer is spot spraying for a particular hardy weed with a particular part of the farm. And then he is using some kind of coverage, very innovative, sheets of galvanized after Maria, and he just solution, obviously glass don't get sunlight, heat, and kills the grasses. That is a smart form of integrated weed management. So the integrated weed management involves mulching, the use of cover crops, selective promotion of crop root health, such as a better fertilizer use and application. In other words, it benefits
We
the
have
crop.
3 minutes left. Sorry to interrupt, Greg. You have 3 minutes left.
And there's even a technology, flame weeding, which is not really practiced in the Caribbean. Soil solarization is another form of weed control. And then there's exploiting the allopathic properties of a little more information on the allopathic compounds that can be released by live plants or when residues decompose, and in general, this results in suppression of weed growth. So these compounds are specific to certain weeds also, and that's not a practice that we use in the Caribbean, but that's something that's been developed in the temperate world to control certain weeds. Smaller crops, as well as mulches, have been shown to release oleochemicals, which will also inhibit weeds. Therefore, the principal goal of smaller crops is to control weeds by replacing an unmanageable weed population with a manageable smaller crop. Colleagues, this is my brief presentation to support the previous presentations that have been made over the past couple of weeks, and to support the presentation show there is the use of bioherbicides on fungi, bacteria, and viruses. Use of fungi, bacteria, and viruses to control weeds. Bioherbicides have many advantages, such as clearing pesticide residue build up in the environment, and effectiveness for control of some herbicide resistant weeds and biotypes. Because basically what we have now is many of the grasses are becoming resistant to the continuous use of the herbicides over the years. And therefore, we need to look for clear alternatives and more specific herbicides. The use of bioherbicides would be great in a country like Dominica, where we have a lot of runoff, high rainfall in certain areas. And as I said, this presentation fits into the previous presentations made, the presentations that will be coming in the future weeks, and is supportive of Dr. Rankin's presentation, which is next. Thank you. I hope I've kept within the time limit, moderator.
Yes, you have, Greg. Yes, you have. You have in a weekend and a week where a few seconds makes a difference. You are definitely within your time. Thank you very much, Greg. The next presentation is going to be by the other R in the double R dynamic duo today. It's Dr. Dale Rankin. And I'm going to quickly read a portion of his bio. Dale Rankin is an applied climate scientist in the Department of Physics at the University of the West Indies, Mona. He holds a PhD in physics with high commendation and worked as the agricultural climate modeling research analyst under the regional track of the pilot program for climate resilience, the PPCR, of which we are all very familiar by now. His research interests are climate and crop simulation, modeling, and more broadly, assessing climate change impacts on the socioeconomics of Jamaica and the wider Caribbean. Dr. Rankin is a member of the Climate Studies Group, MONA, which is headed jointly by Professor Taylor and Professor Stevenson. And of course, all of you know that the Climate Studies Group at MONA has been doing pioneering work in the Caribbean. I have great pleasure in introducing my colleague Dale Rankin to do his thing. Dale, over to you. Over to you, Dale.
Yes, Dr. Maxime, thank you very much.
Okay. And a weekend when the relays are upcoming, don't drop the baton now, please.
No, I will try not to. I will try not to, sir.
Very well, Dale. I'll be back after 17 minutes.
Sure. Okay, let me just see if I can get this thing up. All right, well, first of all, let me thank everybody. Thanks to CARDI for this invitation. Thanks to you, Steve, for the opportunity of presenting. It certainly is an honor of mine. I take it as no mean feat, no mean opportunity. And I just want to say that we have a chance to have some discussions on matters of serious import. And I will say that sometimes we won't necessarily be able to answer all the questions in the session now, but I've been informed that whatever questions you have, they will be logged, and we will get a series of them. For the last presentation, for example, I got a series of questions that I was able to share, answer the questions, and then send the solutions via our dynamic Miss Desi. Okay, so I'm not sure if I have the permission to share my slide. You can let me know. I'm going to try and see what I can do.
You should be a co-host, yes.
Okay, are you seeing my slide now, sir?
Yes, most definitely.
Okay, great.
All right, slide so, view though. It's not in.
Yeah, I'm trying to put it in that mode. Are you seeing it now in that mode?
Yeah, better.
Okay, great. All right, so I'm going to present to you some main findings from a drought tolerance research that we did in sweet potato. The case study we're presenting was done in Jamaica. And what is important about it to remember is that we already know that roots and tuber crops are by nature drought tolerant. And so what we wanted to do was to explore the limits of this drought tolerance. And so I'm going to present, these are my co-authors here. And I want to thank all the sponsors. We have tried to prominently display all of them here. This work was done prior to the PPCR project, I must add. But work on the PPCR project itself was enhanced. So everybody can look on this graph and tell me pretty much that the rainy period is a darker gray and rainfall is king between mid to November. It's interesting because up to the last, I would say up to the mid 1980s, this graph was true. You have a primary peak or a secondary peak in May, June, and the main peak, or they call the primary peak, in September, October, which they call the primary rainfall period. In between, you have a comparatively dry period in July, August, which they call either the midsummer dry spell or the midsummer drought. Now, that is what it was. But in the last 25 years, our study has shown that at least in Jamaica, we get more rainfall in May, June. And this is interesting because in May, June, most of the rainfall that we get is not so much cyclonic rainfall. It's more warm pool rainfall that occurs from, daytime heating and so on. So that's quite interesting. So the timing of this dry spell affects the kind of crops that you plant. If the dry spell is long, you have to plant crops that are more drought tolerant. If the dry spell is less, then you can plant crops that are more sensitive. But the truth about it is that most of our production is limited. Now, here's one of the issues now. One of the problems, Mr. Chairman, and everybody with production in Jamaica and the wider Caribbean is that our yield data is very crudely estimated. So this is a plot of SPI. SPI stands for the Standardized Precipitation Index. The red shows the yield in tons per hectare. sweet potatoes. So on the right-hand side is the axis for yield. On the left-hand side is the SPI. If your SPI is positive and certainly above 1, it is wet. Above 2, it is very wet. Below 1, -1 is dry. Below -2 is extremely dry. And you could look on that graph, even if you didn't understand mathematics, and you could see, especially here in the period post-1992, we were recording record yields of sweet potato when we were having record drought, which makes no sense because the sweet potato weaver, for example, would eat all the sweet potatoes. So we detrended the data. We realized that during this period, the government was just giving a lot of resources for people to go and invest in sweet potatoes. So we realized that we had to detrend the data. When the detrend data is even more obvious, here is a period right here between 94 and 2003, when we have significant drought and we're reporting record yields. So I had to go to the meetings to sit down and find out what was happening. Turns out that, first of all, the yields are estimated across time scales. So January, February, March, all of that is first quarter yield. And then, April, May, June, that is second quarter yield, all of that stuff. And then it is aggregated across varieties. So a ganja variety, for example, and Dr. Robin mentioned that, could produce as much as 30 tons per hectare. But a fire on land variety at the same location could produce as little as 5. So if you take an ensemble mean across varieties, you're really veiling the true potential of the crop. All right, so it is, and then it is also average across locations. So the parish of St. Catherine that has fertile soils, it has dry soils, it has all kinds of different microclimates, is deemed as one production area. So when you take all of that, you will not find any correlation between yield and rainfall. It doesn't matter which crop you take, it doesn't matter which country you take. That is a reality in the Caribbean. Now, why is that important? Because the region is projected by the State of Climate Jamaica and the State of Climate Caribbean to become drier, up to 1 to 3% drier, 40% drier by the end of the century. And by the middle of the century, we're going to see a definite drying trend. And also, we're going to be warmer. So if we're going to be warmer and drier, our production, therefore, will have to be irrigated. Now, so from this data, we couldn't see any trend. So what we had to do was something they call a wavelet analysis. So what a wavelet analysis is, it's an analysis that they use in epidemiology. So they use it to, for example, to highlight the formation of cholera. They were trying to find if there was a link between the number of cases that they were finding on the one hand, and whether or not this was a real relationship. So what we did in this particular case and how we did it, we said, okay, if we're going to do is we are going to have to find whether there's a real link between the two. And what we did was to put the area harvested data, Mr. Chairman, into a matrix and say, okay, if the area harvested has some kind of known cycle, then we're supposed to see a dark red close to a period that tells us what is that cycle. So dark red, you see here, anywhere between 7 years to 2 years. So we say, all right, this is interesting. Then we look across on the power function. The power function on the right-hand side tells you whether it's a strong relationship or a weak relationship. So one, it is inside the cone. So this is a much more fancy statistical relationship than what we call statistical significance. So it is significant, 1. 2, it is red. And 3, it is a strong relationship. But what gave us courage was that the cycle is 2 to 7. Anybody know what is 2 to 7 year cycle in the Caribbean climatology? El Nino. El Nino. When there is an El Nino, it reduces rainfall in the year of onset, and it increases rainfall in the year after onset. And therefore, it told us that what was happening, and sorry for the noise, there's a truck passing my location. So what the data was telling us was that there is an autonomous adaptation that takes place in agriculture. When the rainfall is not capricious, or when the rainfall is not efficacious, I should say, the farmers naturally farm less. When it is good rainfall, they farm more. So we were able to do the analysis. Of course, we use the AcroCrop model, and I told you about this model already, so I want to go back over it again. These are the varieties that we actually use. The ganja variety, you can actually see why they call it ganja. The foliage look very much like the ganja, all right? I can't tell you about the taste because I've never tasted the real ganja, but I can tell you this sweet potato tastes really good. All right, now, this model is driven by what is called canopy cover. So you have to go out in the sun, you have to point a camera above the canopy and take pictures of the canopy. And then it converts that ground cover into percentage area covered. This GCT is not general consumption tax for the Jamaican or VAT for those in Barbados, but it's called the Green Crop Tracker. The Green Crop Tracker was a software that was on the market that I got from FAO. Everybody told me it was perfect. But what we found is that if you look on picture 3, there's a shadow. And it picks up shadow as part of the percentage. We also found that in areas where the ground was almost totally covered. So here's it. The ground is totally covered. The Green Crop Tracker is saying, it is looking at the area that should be canopy and saying this is brown area. So right here, it should be given 87%. What is the Green Crop Tracker giving? 13%. The software that we developed, Automated Canopy Estimator, gave 87.5%. Look at this picture. Again, you see the Green Crop Tracker totally breaks down because it cannot distinguish between when the area when the canopy is so far closed. It gave 1.64%, but that wasn't neither the gap nor the actual canopy. Our software, again, did well. So here's when it gave a large underestimation. Here's when the canopy, the GCT totally breaks down. And the shadow, of course, is a problem. So we were able to write a paper. We got best best publication by the Office of the Principal for this because we were able to outperform 8 other existing estimators on the market. So what did this do? Well, turns out that when we were using the GCT, and I said before, canopy cover is important, we saw that we could not properly guess what the model was doing. We couldn't properly represent, Mr. Chairman, what the model was doing. However, when we use ACE, we were able to get a better fit between what was simulated in terms of canopy cover and what was measured. So the dots with the error bars here represent what was actually measured, and the solid line represent what was irrigated. How did this manifest itself in terms of actual impacts on the ground, both for climate smart agriculture and agriculture general? Well, here you see, Mr. Chairman, in the top column, top left hand here, I'm showing you Devon in Manchester in 2013. You can see that the canopy cover was almost bang on perfect. Once the canopy cover was well measured, you go down, the biomass is also excellently measured. The same is also true for Ebony Park. Ebony Park gives not such a great simulation, but that's what you want to see. Sometimes it's overestimating, sometimes it's underestimating because all models are wrong, but some are useful. And then you see that we were able to capture in general what's happening on the ground. Most models have a 10% error. So if you measure 20% in the field and you get a ton, if you measure 20 tons in the field and you get 18 in your model, or you get 22 plus or minus 10%, that's perfect because the model has an inherent error. So what we found in general in this particular case, that the sweet potato was an original contribution. Nobody had done it in the world before. There was excellent agreement between what was measured and what was simulated for canopy cover. The model actually shows that it's very good skill at both locations when it was actually doing simulation of biomass. And what was critical for us, when canopy cover was well simulated, so too was biomass. The same thing is true for yield, and everybody will be happy to hear that AquaCrop does well as well. So if you get the canopy cover right, you also get the yield right. So that's perfect. Now, why is that important for you guys? Because you have a model that you can go in the field, you can take your picture. Now, the software that we have developed is even more accurate because it doesn't matter whether you use a Fujifilm camera or a Canon camera or you use a cell phone. Once you upload the image to the software, you get the digital digitization up to 2 decimal place. We have a problem on the US supercomputer because what was happening, I could send you the link, you could upload your photos, and immediately you would get back a thing in seconds. So it was the fastest on the market. Unfortunately, there's a space issue now, so it's not online right now. However, Dr. Andre Coy, who was the team leader for this particular part of the research, and myself, we avail ourselves. If people have images and they want to get it done, we can process it. Takes a little longer though, because of course, in this particular case now, we have to upload the thing manually. What we want to do for the future, Mr. Chairman, is we want to automate the data. So what would happen is that if I go in the field and I have randomized complete blocks, which means I have 4 varieties and I plant each variety 4 times, I would have to manually tell the model that picture number 1221 is ganja, picture number 1222 is fire on land. But we believe that we could do a model training system where we could have a camera on a trellising system. And we train the camera that every time you see this foliage that look like ganja, this is ganja. We go through the field in record time, it would take all the ganja, it comes back to a starting point, it takes all the fire online, it comes back, it takes all the yellow belly, and that way it captures it much faster. We experimented with drone technology that worked pretty well, but the drone technology, you really have to have a very skilled person flying the drone. We almost lost a drone in strong wind as well. So I would recommend the trellising system if you really want to do it. We also found that for cassava, interestingly enough, that irrigation does not always improve yield. Here's a summary. I have 7 minutes to go. Here's a summary, Mr. Chairman, of what we found. So what was measured in 2012 was 14 tons plus or minus 6.3. What was simulated is 10.3. A deviation, which means measured minus simulated, as expressed as a percentage, was 29%. For the rain-fed, it was 22%. In 2013, we saw, this is for yield now, for yield, in 2012, we had 11.2. Some of you are reading faster than I am, so I won't go through all the figures. In 2013, you saw that we got a much better fit between measured and simulated. And again, I said there's an inherent error of 10%. So if you're getting 20%, that means you're in error 10%, which is pretty good. For yield, and this was published in one of our papers in the Agronomy Journal, Mr. Chairman, you see our yield was 6.7 that was measured, and our simulated was exactly 6.7. So we were bang on perfection on that one. So that was excellent. And we were really very happy about that. What can we say now about the model performance itself? There's this article called E. E is not for egg, but E is in an expression. Some people use D statistic. It's a measure of how the model performs at every point in your data point. So it compares, so if you have 5 or 6 measured data over the crop life, and in sweet potato, this is a 6-month crop, it measures what is the difference between the measured and simulated for every point and tells you overall what your average is. So the closer you are to +1, the closer you are to perfection, if you can just bear that in mind. All right. So you could see that at Devon in Manchester, in the first year, we had negative values, which wasn't very good for canopy cover. We did much better in 2013. Ebony Park, we didn't plant Ebony Park in 2012, wasn't so bad. Biomass, 0.7. Rain-fed was bad because the model doesn't do very well in dry conditions when it's very poor. But we got 0.79, 0.74, 0.39, 0.89. 0.89 is excellent, considering that rain-fed at Ebony Park. And the yield values are there for you to see. So that wasn't bad at all, Mr. Chairman. Now, what about climate change? Well, we looked at something from the data that we had. We said, okay, what if we chose the worst possible year, the lowest amount of rainfall? Sweet potato needs at least 500 millimeters of rainfall. What if we took the driest year and say, okay, this is 74% below what the mean rainfall is? And what if we took the hottest year? So under climate change, since we never have the future data, what would happen under dry and warm climate? In rain-fed conditions, irrigated conditions, you can see that the yield change was negative, all right? When you factor in accelerated CO2 or increased CO2, Mr. Chairman, you realize—
Sorry to interrupt there, but you have 3 more minutes.
Sure, thanks. Thanks, Mr. Chairman. What we found here is that essentially when you factor in increased CO2, you saw that the negative values turn positive, which means that even in warm, dry conditions, the increase in CO2 will have a beneficial effect on yield. The extent to which that will happen, we're not sure. When you look on the cool and wet scenario, though, you notice something is interesting, that in general, the cool and wet conditions favor, increases above the baseline. However, what we notice is that when you have cool and wet plus irrigated, you don't get a net benefit. Look at this. In the first case, you have 6.8% increase. In the irrigated, you have 5%. In the second case, you have 18.7. In the second case, you have 16.8. 15 versus 13, 28 versus 13. What is that saying? Because sweet potato is already drought tolerant, when it has conditions that meets its evaporative demand, giving it irrigation does not enhance yield. All right. And so this graph is just showing you 2 things. In the warm and dry climate, you can see that the rate of increase of yield and versus irrigated versus rain-fed is less steep But what you should also see, and probably those who are not maths inclined would not see, sorry, Mr. Chair, but what you'd actually see is that as you move forward in time, the slope of the graph reduces, which tells you that after a while, the beneficial effects of CO2 are no longer efficacious. Finally, how do we expand this to something else? Well, I'm happy to tell you that we have invented an app called Accept Agri. All right, Accept Agri grew from a project proposal that I developed, my brainchild, Agriculture Climate Change Evaluation for Production Transformation Resilience. Mr. Chairman, I don't have enough time by my watch. I have 1 minute. So let me just see if I can bring it up in exactly 1 minute. So this is what it looks like. The portal comes up. Are you seeing my slide, Mr. Chairman?
Yes, I am.
Okay, so this is what the portal looks like. All right, you can go down, read about the information there. Essentially, you can go inside the pool, the project, you can select a location that you want. When you select the location that you want, for example, you come here, if it works, all right, you can click on a location, you select your crop, you come over this side, you select the crop, in this case, you have dasheen and cassava. Once you select the crop, you select the cultivar that you want, all right? When you select the cultivar, you click in a location, it auto-selects your location. When it auto-selects your location, it selects your soil type and the closest weather station. And then boom, it gives you a simulation, something looking like this. If the simulation would come up, it gives you a simulation looking like this. All right, you can see your yield, you can see your biomass, and this is my last thing. It shows you what your yield potential can be. Now, considering that what I mentioned before, that no country has real yield data, we think this is a gold medal that is waiting to be unfurled. We think if we could get further funding, it would help us because we could increase it to not just Jamaica, but other Caribbean islands. We could have a range of other crops, but it needs a lot of funding. All right, Mr. Chairman, I have 2 seconds left, and with that, I end my presentation. I thank you very much.
Thank you very much, Dale. What a pitch. Okay, so colleagues, you're familiar with the format. We have deliberately provided you with information that you can interrogate. This is the idea behind this webinar series, not just to present the information, but to give you an opportunity to. The procedure is quite simple. Have a question, raise your hand, and our expert team in the background will unmute your mic. If not, and you want to type in your question, please type it in the question and answer icon, and your questions will be presented to the panel. I will do that. I think I'm able to read those questions. So let's take the first question. Oh, it's not really a question, it's a comment. And yes, we will be providing those who require with a certificate of participation so that you can get clearance with your supervisors, etc., for the time you spent here with us. So that's a no-brainer. That's cool. Any other questions?
Hi, good morning. This is Dr. Aline from UWI Caper Campus. Can you hear me?
Yes, I can.
Okay, great. I would like to thank the presenters this morning. I thought that the information was pretty on point. And I was quite interested in the last presenter on the Accept Agri Act and also on the ACE. My question is about diseases, because while root crops are critical for future development in terms of drought tolerance and so forth, does the act, for example, measure the, because I know some of the disease models use brownness and greenness as a measure of extent of disease, for example. Does the Accept Act take that into consideration, and how is that working? How are we going to get accurate data in terms of yield based on disease if we can figure that into the act? Can Dr. Rankin answer that?
Dale, please.
I never got the question. Can the person just repeat it?
The question is about disease. Plant lesions, for example, brown areas in green areas. and the app measure yield accurately when diseases areas that are missing in a scenario like that?
Sure. Dr. Aline, nice again. Nice to hear you. Well, first of all, thanks for your question. All right, I should point out, Mr. Chairman, forgive me. I'm not trying to elongate my time in a cheeky way. I should say this, that as it right now, the app is based not on AcroCrop, but it's based on the more difficult model, which is C-SAC, and these are pest-control mechanisms in it to deal with the issues of pests. So it is a more sophisticated model. What we want to do is to make the first multi-model platform in the world, which means there are some crops that are in AcroCrop that are not in D-SAT. For example, D-SAT does not have a sweet potato model. AcroCrop does. So if you were to go on the planet, type in sweet potato, it would just naturally default to AcroCrop. There are some models, there are some crops that are models. And in that case, we'll give you an option. So to answer your question, Dr. Aline, yes, we have something that can deal with pests. However, there is no funding right now to incorporate that. And unless somebody is willing to fund that, we will not be able to give you that functionality.
Okay, thank you very much. I'm quite willing to work alongside you guys for the Barbados models in terms of pest and diseases in root crops, because that's, you know, is where my interest lies. So later, and hopefully in the future on the funding and so forth. Thank you.
Thank you, Doc.
Further questions? There are no questions in the Q&A.
Steve, are you able to hear me? Ian has a question. I just gave him permission to speak, so you can go ahead and allow him to ask his question.
Please go ahead.
Yes, Steve, good morning.
Ian, this is the first time I'm giving you permission to speak. That's great. Enjoying this.
Yeah, I think so, yes. Question, I joined the meeting a bit late because I was under the impression that we were starting at 11 a.m. Barbados time. But both Gregory and Dr. Rankin's presentations were quite good. But I have dealt over the past 45 years with insect and white pests, particularly on a number of crops, including things like the root and tuber crops, particularly sweet potato. And as you might know, with the increasing climate change, the incidence of dry weather, these crops, when they're under drought stress, tend to be much more susceptible to the sucking pests. I'm looking at things like white flies, like lace bugs, and red spider mite. Is any work going on in the Caribbean on looking at what varieties, say for example, in sweet potato are showing some degree of tolerance or resistance to these sucking pests, and even our old enemy, West Indian sweet potato weevil?
Before I ask Dale and Gregory to comment, Ian, I remind you that we're going to have a full entomologist session next week. We're looking at integrated pest management, and there'll be people maybe a bit more qualified than Dale, definitely, to speak about it, right? But I'll give Dale and Gregory a chance.
Let me just briefly say, Chairman, well, you rightfully said that, and I indicated in my presentation that the pest and disease management was going to be handled in a webinar to come in the next couple of weeks by Dion Clark-Harris. However, with respect to the sweet potato, we have not seen any new pests in our drought tolerance studies. Work has been done on the weevil, both in St. Vincent and in Jamaica, very detailed research, and there are packages of biological and conventional methods of treating both pests, both types of evil. Cassava, I know recently, and there's a paper being published by Dr. Allen from Cave and myself on cassava elongation disease, which is something that is, I don't know how new it is to the Caribbean, but it's there. And that's something we've been observing in some of the works we've been doing as a new problem in cassava. I suggest that you follow the next week, I think, the webinar presentation on pest and disease management. Dion is probably not on right now because she's in the field. Otherwise, she probably would have responded directly to your question. But you're right. There's some increase in pressure on the plants because on the drought, they do become susceptible. I must say something about cassava, and I think Dr. Rankin indicated that cassava didn't really respond to irrigation in terms of yields. Cassava has good drought tolerance when it's established. So if you establish cassava for 3 months and it's fully established in irrigation, it can withstand most drought tolerant conditions. And why cassava is so widespread in Africa, where you have a lot of drought and years without rain, is because it has good drought tolerant characteristics. And those bred now can really withstand the rigor of drought and pest and disease. So these are my comments to your point.
I hope that helps, Ian. If I could also add my tuppence, I know that we are currently, when I say we are a team headed by Dr. Yard and myself, looking at invasive species on NESAP for Barbados. And we are going to be looking at the increased waves of pests that are coming in of IPPC, etc. So I hope, Ian, you'll stay with us when Dion and Dr. Robinson do that session on integrated pest. I'll bring it up again then.
Mr. Chairman, I know I'm the least of the apostles in this regard, and I noticed that you have closed the question without my intervention, but that's fine. I mean, suffice it to say that there are some studies that have emerged. I will say this, that we have not done a lot of work. That's the real truth. I know some stuff has been done in Jamaica. We did some studies ourselves. In fact, I'm actually in the final stages of publishing a paper, and I can't give the results of that paper now before it is published. But we would need more work, though, Steve. That's the reality of it. Some of the stuff that we have seen in our region, I can't talk about Africa, are a slight variance with what I hear a lot of people say about African studies. Our studies are very different. Our results, our climate is different. Our management practices are different. Our soils are different. And therefore, the interaction between soil, cultivar, and management gives a different result. But I wouldn't want to appear to be contradicting my esteemed colleagues. I would want to wait until we have more studies and we can definitively establish a trend before I say anything on that.
Okay. Thanks a lot, Dale. I will get back to you shortly, but I'm going to take a question from the Q&A from Fires. What weather data or climate data, if any, would be required to run the Accept Agri app in Trinidad and Tobago? Did you get that, Dale?
Yes, Mr. Chairman. Mr. Chairman, okay, so Accept Agri is based on a model called, a software application called the Decision Support System for Agro-Technology Transfer. Nobody remembers that. Everybody said DSAT, DSSAT. Now, you won't see the DSAT model. We run the DSAT model in the background at UA. We just give you an easy interface. But obviously, we'd have to get the coordinates and all of that for the soils and so on of Trinidad to then put Trinidad into the software package. Make But we have to demonstrate proof of concept now that it can work for Jamaica, it can work for anywhere in the world. The weather data that is needed to run the models that are in the C-SAC software are namely these. 1, minimum temperature. 2, maximum temperature. 3, solar radiation or sunshine hours. 4, relative humidity. 5, wind. And 6, rainfall. That's the ideal list. Now, if you don't have all of that, the minimum you would need is minimum temperature, solar radiation, and rainfall. You must have those 4. If you don't have solar radiation and your Met Service can give you sunshine hours, that can work too. But yeah, you need an absolute minimum of 4.
Thank you, Dale. The next question is from Nikolai McLean. Would you get a good yield of cassava if you plant it in big bags?
I think that question is for Greg.
I would pose it to Greg.
Can you repeat the question? Will you get a good yield of cassava if you plant it in what?
In big bags.
In bags.
Yeah, as a climate smart.
It's big. Okay, can you get a good yield of cassava if you plant it in big bags?
Yeah.
I suppose it depends on the size of the bag.
The root in that bag.
And then what you use as a media in the bag, which is probably more important. It's like planting cassava in a big drum. I think what would be more important is the actual medium, what you put in, whether it's a mixture or sandy clay loam and you put in some compost in it and stuff like that. I think the media and not so much the bag is the important factor.
Fair enough, Nicola. I hope that helps.
Mr. Chair, can I say something?
If you must, I don't really
All
like
right. I just a quick one. I think it would depend on the cultivar itself. Just our quick experiment with 4 cultivars. And Greg would be able to attest this with me. It depends. If there's a variety called C119-5, you couldn't grow that in a bag at all. It spreads out all over the place. It would tear up the bag. There's a variety called BRA 383 that doesn't branch at all. It goes straight up. That could go in a bag. So it depends on the cultivar. That's all I want.
Yeah, okay. Dale, for once you made sense. Greg, you want to confirm that?
Thank you, sir. Thank you, sir. At least that makes sense.
Well, as I said, you can take the bag and look at it as a drum, a big drum. So obviously the bag, it is a fertilizer type bag, the roots are going to put pressure on it. And as Dale says, it's going to repart over time, all right, with soil in it. But I think I'd advise the individual who asks the question is to grow it in drums, all right? And as Dale said, you have different architecture. Cassava has different morphological architecture. There's some that branch profusely. You probably wouldn't want to put that in a drum or in a bag. Those that go straight up with limited branching, but all these are bred for specific growing situations. So I'm assuming that could hold. But my recommendation would be to grow it in a drum. Like we grow yams in drums, we grow sweet potato in drums, we grow sweet potato in bags. So many of the root crops can be grown in a containerized fashion.
Okay, thanks for that, Greg, and limited thanks to Dale. I have a very interesting question in the Q&A that I'm going to read now from Shanyon Clark McPherson. Good morning, everyone. Thanks for the information. So what do you think about the sargassum weed for mulching and for weed control? Either Greg or...
I attended a sargassum conference at Muna. Very interesting conference. And there is work going on. I know a colleague of mine in St. Lucia was using sargassum as a mulch. and also incorporating it, dried sargassum as a form of fertilizer. One of the concerns was at the time of presentation that not enough work was done on the actual chemical composition of sargassum, fresh and dried, and to look at the residual effects in the soil and as a mulch. So it has been used as a mulch, and I know that some work has been done in St. Lucia, but I don't think at the time of presentation, the results were conclusive. So I wouldn't like to report on it here.
What I can promise to do.
But I could put that question could be put to Andrea Vieira, who's now at UWIN Antigua. And I know she was one of the scientists, Dr. Andrea Vieira. She was one of the scientists working on that. So she could give some feedback in terms of clarification to the particular participant. So make a note of that.
Yes, I was also going to suggest to Shania that I will contact a couple of my colleagues in Barbados and St. Lucia who are actually working on the use of sargassum, including some of the proprietary products that have already been made. So I'll put that into the general conversation later on. I'm going to ask Desi to remind me to do so.
Steve, just to bring your attention to Dr. Arlene, she has her hand up. I think she wants to ask a question.
Oh, please, you're the one doing the releasing. Don't let it appear that I'm keeping Dr. Arlene away, please.
Okay, I just put my question in the chat anyway, but my question was for Greg. And Greg said something about clean planting material. I think is critical and something that we've been talking about for years in terms of crops in the region, especially sweet potato and cassava and yams. Greg, you talked about the fact that we do have seed banks. We do have, we should be going to these banks and to CS and so forth for clean planting material. But my question is, you listed about 3 or 4 of these the tissue labs, because we have one in Barbados and in St. Vincent, et cetera, et cetera. Are they developing standards? Are they talking to each other to produce standard protocols for the release of these clean planting materials to farmers? Because like you identified, there are systems in place that show that when you have on-farm or particular farmers who are responsible for developing clean plants for then distribution island-wide, that is where we should be going. So what is in place for the TC labs and the seed banks to develop these standards so we can get a clear idea of what is the standard throughout the region? Can Greg answer that?
Well, okay. We have these facilities in the Caribbean, and they're at different stages of, for want of a better word, certification. There's different levels of staffing, equipment, and skill sets to store, clean up, multiply, and redistribute. The facilities that I'm aware of, and I noticed Omaira in the participants chart. I don't know if she's still there, but Omaira has a good knowledge of the laboratory systems around the region. So SRC, the lab in St. Vincent has been compromised because of the volcano. There's a lab in St. Lucia. There's some facility in Barbados. The idea was to, as you rightfully said, standardize the processes of getting not only material from the CGR institutions, C-SAC, IIA, which we've done over the years, but to getting the material based on characteristics clearly defined by the markets, whether it's for local consumption, regional, or international export. Secondly, the methods and processes for the laboratory work itself, of reculturing and multiplying. These are standard techniques that I don't think we are not aware of. I'm not saying that they're should, and there's still sometimes some levels of contamination in the various labs. I think the problem is because the root crops are vegetative material, their distribution pattern is between ministry stations, farmer to farmer. The literature and the research has shown clearly that after every 2 to 3 years, it is advisable to go back to the lab for clean planting material to recirculate into the farming system by having special facilities for propagating and growing out for multiplication purposes and distributing. We did some work and we've identified drought-tolerant varieties, the ganja, for instance. We'll be able to cover it molecularly field work. So it would be good for one of the labs, preferably the SRC, to select out and take that variety through the process. The reason why those things are not done, I really don't want to articulate that in this forum, but we have the skill sets, we have the laboratories, some of them need upgrading, and once we get those things in place, we need to perfect the methodologies for planting material, clean planting material distribution, because the lab itself will build under the CSC specifically address of root crops.
Thanks, Greg. I just kind of reiterate how important the clean planting material is, and that we need to have a program in place to address that through the lab systems right now, because I think the labs are there. We need to put emphasis on that. So thanks.
Agreed. Doc.
Okay, thanks.
Let me take another question from Ms. Clark McPherson. Sweet potatoes tend to bear here healthily in one soil, the ashy soil. Okay, there was a comment really from Shannon speaking about the fertility of the ash-laden soil in Montserrat. I don't know if you want, either of you wanted to comment on that comment.
But the ashy soil, I'm not quite sure what that description is, but it sounds like it's kind of a sandyish, loomish kind of soil.
It's volcanic soil with ash from Montserrat.
Ashy soil, I'm not quite sure what the physical makeup of that is, whether it's a sort of a sandy clay loam soil, because I know in St. Vincent you have this kind of sandy, gravelly soils, not from this eruption, but from the last eruption of that very Soufriere Hills volcano in what, 79 thereabout, or whenever it has erupted previously. And it provides over time a sort of fertility to the soil. So there's no accident why St. Vincent is a huge root crop producing and exporting island, especially to Trinidad and Barbados. But they have this sort of a sandy, ashy, kind of fine gravelly soils in some areas in the Mesopotamia Valley, and so which are very conducive to root crop production, especially sweet potato, in some instances yam.
Okay, thank you, Greg. Any other hands raised? Questions? Desi, I have not been going through the chat. Have you been going through the chat looking for questions? Because I haven't.
Yeah, most of them ended up posting their questions in the Q&A section. So we have covered majority of the questions.
Okay.
Yeah.
On the subject of climate smart adaptations and protected agriculture, Greg you mentioned, you started to talk about growth of root crops in containers. What has been your experience Caribbean-wide in the use of containers for growing root?
I saw a very good example of sweet potato grown in containers at Booths in Jamaica. And in that instance, they used growth in containers for planting material. So the focus was not tubers. I have seen sweet potatoes grown in huge drum type containers. I don't know if these blue drums they use in the paint industry, so they cut them in half and they put sweet potatoes in that, and they do pretty well. Yams grow well in drums, all right? So sweet potatoes, yams, cassava, because of the nature of the tuber, they can be very longitudinal and spread out. It depends on the genotype also. You can't grow in drums or bags, but I haven't got any experience of cassava in drums and bags. More of sweet potato and yams, especially yams. Yams are grown in drums very often, and sweet potato also. So there's no problems in doing that.
I was particularly concerned in terms of some of the nutrition security suggestions for urbanized areas where people do not necessarily have backyards, but they may have, areas where they can have 20, 30 bags of a particular crop growing. So I just wanted to hear your experience, because I have seen several of those do quite well, actually. Any other questions on climate smart adaptations? Greg, as someone who was in charge of the root crop, the root and tuber aspects of CARDI's research, can you give us any additional details on the point I raised in the very first webinar about the use of planted material from climate analogs, like in the Pacific, as with the dashing cultivars that CARDI introduced to the region? Could you give us some background on that?
All right. We thought for a long time that the genotype base for dashing in the region was too narrow. And like the Pacific, where they had taro bacterial blight wipe out the crop completely. And that led to the Australians supporting a huge program under the International Network of Edible Aroids to collect genotypes from all around the Asian basin, Pacific, and they developed a huge breeding program, and they came up with many genotypes that were resistant to bacterial blight. We became part of a project purely to address our narrow genetic base, and if ever bacterial blight came to the Caribbean. So we brought in 42, 45 genotypes from the lab in the Pacific. We morphologically characterized them, and then we selected out and did farmer participatory trials. While we did on-station research to validate the 10 or 15 that we selected out from the initial single row observation trials. And we were able to get 2 genotypes, and St. Vincent market tested them in Trinidad, and they did very, very well on the market in Trinidad. So we also were able to, not specifically doing drought tolerant experiments, but our experiments were in the field. That year, I can't remember what the year it was, whether it was 2009 or when we had this intensive, prolonged dry spell in the region. and everything died out. Taro died back, sweet potato died back, and in that period, 3 to 5 of the genotypes remained green right through while the others died back. And we identified them for future drought trials. All right, so we have in the lab in St. Vincent about 40 genotypes. In the lab in St. Lucia, probably about 20 genotypes from what was brought in from the Pacific. I've identified about 3 for commercial production and export, human consumption, and about 5 that have good drought tolerant characteristics. One of the issues we had was when this genotype, we were very cognizant of the fact that St. Vincent has a thriving export dashing business to Europe and the Eastern Seaboard in the States. And the ministry indicated that we have to be very pragmatic in how we approach taking any of the introduced cultivars to commercialization, because when you have a tried and tested market, you have to ensure that if you introduce anything new to it, you don't affect your branded trade that's going on. So that's where we were. We were doing some market testing, and the feedback I know from Trinidad was pretty good. You know, Trinidad grows taro in many instances for leaf and not tuber. So we have to ensure that those 2 varieties tested on the market were ready for tuber consumption. So that's when I left St. Vincent, that's where we were.
Thanks, Greg. I mean, this is something that I'm particularly concerned about. Once we talk climate smart agriculture, this heavy reliance we're going to have to place on both drought tolerant and salt tolerant varieties is going to come to the fore. And I was really, really pleased, as I've been reporting for years, when a Vincentian farmer came up to me and said, you know, that these new varieties were God sent, you know, and that the market acceptance in Trinidad was exceptional. But I take your point. ECTAD has a thriving, you know, business going on in the Eastern Seaboard and in the UK, and you wouldn't want to introduce a new variety to an established market. From a climate smart point of view, I think it was really, really important that we developed, we tested some of those varieties.
I think also I would like to mention, we did participate with trials. This was a request from the project. So while we were doing this state-of-the-art research on the stations, the farmers had the technology and were doing single row observation trials on the row supervision. They were using their management system. So we were collecting that data from 25 farms in the Richmond area in St. Vincent on these genotypes, and the farmers produced very important guidance in terms of consumption and what they like and didn't like. We also did organoleptic tests, so we knew clearly what the pattern of consumption was. But you know, as Dale always laments, we don't have the funding to do research. For instance, we did some work with the accessions in Jamaica and clearly showed drought tolerant ganja one of the accessions. Dr. Alan Barrios did the molecular work and was able to show ganja has salt and drought tolerant genetic characteristics. We're supposed to do that for all local commercial accessions and see what we have. We could use them in future breeding programs. But we don't have the resources to do that kind of research.
Well, so fortunately, Greg, that's where people like me come in that now that we are getting this climate smart agriculture thing really established, standardized, getting the MRV data that we would need, I can assure you that there will be climate finance coming for all of these types of activities. And again, once more, with a final plug for ECO, that Ag-Ready project, that readiness project that has been conducted in 6 countries, is going to put on a stamp in terms of dating the whole climate smart agriculture approach in the Caribbean and the fact that we are low emissions producers, etc. I'm looking again in the Q&A and I'm not seeing any, so can we take one close-off question before I ask both presenters, excellent presenters, to give 1 minute wrap-ups. So I'm not seeing any hands raised or questions. So can I first go to the list of the apostles? Can I go to Dale first? Dale, can you just give us a 1 minute wrap-up to day 5 in this double week that the Caribbean is experiencing?
Well, just quickly say, Mr. Chairman, that I wish the euphoria that we have for the Olympic Games would spill over into applied research, and that all those persons who are involved in extension services and funding and so on would allot and pay great attention to applied research. Let's not make our food security dependent on somebody external. Let's invest in it and put our money where our mouths are.
Thank you, Dale. Greg?
Yeah, I think from the first presentations, climate smart agriculture is an all-encompassing process. It includes GAP, farming, organic agriculture. And I think on the organic, from what I've been looking at and reviewing and reading, it doesn't have to be certified. But once the organic can help in sequestration of carbon and so on, then we'll be doing fine. I think the concept of survivability probably not getting there, but climate smart is to allow us to survive and also take care of ourselves. As Dale says, we shouldn't be dependent on other people for our food security. And I think we got to get that message across, and I think this webinar is slowly getting it across. Climate smart is not about just producing crops and trying to do the best production system. It's an all-encompassing process that deals with the welfare of the farmer, his family, the environment, and the processes we use to produce the commodities.
Yes, it is to survive and thrive. We don't just compete to participate, we want to compete to win. So in a week when we are hoping to spread that euphoria, as Dale so eloquently mentioned, the euphoria of the double, so a similar type euphoria in supporting the talent, the very, very raw talent that in some cases that exists in the region in terms of our climate smart agriculture applications. I want to invite all of you to attend the 6th issue of this webinar series, when we are going to start, just as in the Olympics, seeing the superiority of the female contributions to this entire topic. From tomorrow, we will be hitting a 400 of just female interaction. We have heard from a lot of these males the last 5 issues, but I can assure you that starting tomorrow, 10 o'clock Jamaica time, 11 o'clock Eastern Caribbean time, we're into a brand new era of female domination as we've seen in sports in climate smart agriculture. I thank you all very much. Being with you tomorrow. Bye.